All right, I think let's get started. I hear this clock is slow and this clock is correct on the computer. Good. So good morning. Welcome to hybrid seminar. Thank you to those of you who came in person. It's really good to see you. It's my pleasure today to introduce Dr. Logan Spector. And so Logan is a friend and colleague. I'm going to try to do this without notes. Logan got his PhD from Emory in epidemiology and then came to the University of Minnesota to learn about childhood cancer as a postdoc, stayed on as faculty, and now he is professor and division director for epidemiology and clinical research in the Department of Pediatrics. So as you can see by his title, which I'm not sure if the title was to try to get the Tversky group here or not, but Logan is very creative and he's very good with titles, bringing such things as rare to well done, the epidemiology of Ewing sarcoma. The 10,000 families name is also attributable to him, which was a blessing and a curse. And he is also, beyond just words, very creative, bringing you things like the D2D building. So doing research on a stick was Logan's idea. And he studies a variety of childhood cancers, leukemia, osteosarcoma, hepatoblastoma, and today he's going to talk to us about Ewing sarcoma. So thank you, Logan.
Thank you. Yeah, actually, as I was coming in, I was thinking about how I first became interested in Ewing sarcoma. So when I arrived here as a postdoc, my late mentor, Julie Ross, assigned me to first task, a classic postdoc task, to write a chapter for Schottenfeld and Frau-Maney on pediatric cancer. And I arrived here knowing about just acute lymphoblastic leukemia, which was my dissertation. But as I was doing this like self-directed crash course in other childhood cancers, I became pretty fascinated by the bone cancers, osteosarcoma and Ewing sarcoma. I like to call them twins where both are evil, but in different contrasting ways. Osteosarcoma has one of the most complex genomes of any cancer, whereas Ewing sarcoma is remarkably homogeneous, with 90% plus having a single driving mutation that seems sufficient to cause the cancer, and yet both have a very poor outcome, which has been very resistant to improvements in treatment for the last 30 years. I also am pleased with my titles, and so I've given this one 2 titles. The real title of this talk is Ewing sarcoma, the zebra with polka dots. I was too embarrassed to put that out in public, but you all can have it now. And the reason I call it that is this. There's a saying in medicine, when you hear hoofbeats, think horses, not zebras. And Ewing sarcoma is the ultimate zebra because no clinician is going to think of it. They will go undetected for many, many months. When they finally find it, it's probably a once in a career thing for the primary care physician. And so today you're going to get a crash course in Ewing sarcoma. It's not a cancer that most of you will ever encounter, scientifically, or if you're a clinician, clinically. But it's a fascinating story, and so I thank you for coming along for the ride. I'll talk about tumor biology a little bit, the descriptive epidemiology, which is remarkably interesting, the small amount of analytic epidemiology, the genetic epi, which we're moving into, and then talk about fantastic collaborative project with Bo Weber and more, and then our future directions. So back to history. James Ewing is one of the few people who gets a cancer named after him, along with whoever Wilm was, and whoever Hodgkin was. And then I feel really bad for whoever discovered non-Hodgkin lymphoma, because their cancer is named after the absence of being Hodgkin's. So James Ewing was described a diffuse endothelioma of bone in 1920. It was later named after him. I don't even know who the naming authority is who decides that some cancers get to keep a name, but It turns out he was important for many other things. He founded Memorial Hospital, now known as Memorial Sloan Kettering, and he helped found both AACR and ACS. So I suppose that, getting a cancer named after him is kind of a reward for being an important historical figure. If you'd like to read about the biology and clinical aspects of Ewing sarcoma, there was this fantastic review in New England Journal a couple years ago, with the first author being the Italian king of Ewing sarcoma, Niccolo Righi. This just shows you what it looks like under a microscope. It is a small, round, blue cell tumor. And I'm really happy that the pathologist decided to give it really easy words, so I know exactly what those cells look like. And we have known for 30-ish years that there is a single driver mutation that is almost pathognomic for Ewing sarcoma. In other words, if you have the EWS FLY1 translocation, then that is itself diagnostic for Ewing sarcoma. You don't see it in other cancers, and so on. The other 10–12% of Ewing sarcoma is EWS with other fusion partners, and not so much. We don't here do too much research into that because EWS FLY1 is the major one. There is a lot of work looking at, what is it that EWS FLY1 does? And, not surprisingly, immediately upon its discovery, much like contemporaneously when they discovered P53, people thought we would try to target that protein. And I'm not a medicinal chemist or anything along those lines. I don't really know why EWS FLY1 and these other translocations seem untargetable, but they have. And so a lot of attention has moved on to downstream targets. So EWS FLY1 enacts a pernicious program of transcription, and we are chipping away at how it acts. And then the project that I'll get to later with Bo et al., we're really trying to isolate what are the downstream genes that EWS FLY1 affects, and wonder whether we might be able to intervene at that point. The other thing that you need to know about EWS FLY1 is it's remarkably toxic to normal cells, and so that has hampered the creation of model animals. It also makes it difficult to study in vitro. Although I've never seen anyone actually document this, it's just well known in the Ewing sarcoma community. There's only a few cell types that will tolerate EWS FLY1 long enough to study. And so that informs the project that we'll tell you about in a little bit. I mentioned that it's one of the more homogeneous tumors in that it has a single driver mutation, EWS FLY1, that is present in nearly 90% of the tumors. It's also, like a lot of pediatric cancers, fairly silent otherwise, mutationally. And so where are we on the Ewing sarcomas? Right here. So middle of the pack for childhood cancers, but the childhood cancers as a whole are, except for high-grade glioma, are much less mutated across the genome than adult cancers. However, there has been, some research into cooperating mutations or mutations which enhance oncogenic activity. And so, I still have to get my magnifying glass to read and understand these oncoprints, but the title will tell you. The aggressiveness is modified if there are STAG2 or TP53 mutations. So those are also a matter of study. And here's the Kaplan-Meier curves showing a difference in survival depending on mutational status. And then there are international working groups which have tried to use molecular biology, patient characteristics to try and stratify patients. And, you know, this is fairly typical in pediatric oncology and maybe adults, but what started out as a simple question, how do we stratify patients, is now a extremely complicated algorithm. But the other thing that we found, we as the field have found, is that if you see circulating tumor DNA at diagnosis, then that also is affords a bad prognosis. So in the therapy space, there's a lot of prognosticating and a lot less actually improving the cure. All right, so that's it for the clinical aspects. Now I'm going to tell you why I call it the zebra with polka dots. It's because Ewing sarcoma has just a number of unusual features that make it supremely interesting and very different than a lot of cancers. And so I already mentioned that a single somatic mutation, EWS-FLI1 translocation, is sufficient to cause the tumor. And I would add that it's unusual tumor head homogeneity. I'm having a hard time thinking of other cancers where 90% of them are driven by a single mutation. And then there's the exquisite cell types specificity for EWS-FLI1. I already mentioned osteosarcoma as the evil twin, and they often are contrasted because they're both considered bone tumors. Osteosarcoma is very clear that it starts in osteoblasts, and most of the tumors happen during puberty in the long bones of the lower limbs, which is where you get most of the growth and height during puberty. And so there's a very clear association between growth, the cells that are dividing during growth, and the occurrence of osteosarcoma. Ewing sarcoma, oddly, has a peak during puberty, but the distribution of tumors is more even. It's almost proportional to the amount of bone across the body. And then oddly, it's not clear that it's called a bone tumor, but it doesn't produce osseous material, and it's not clearly a cell that is involved in bone growth or metabolism. In fact, there's some speculation that the Ewing sarcoma cell of origin, and I did not have time to stuff in our investigations into this talk, but there's some speculation that the cell type that causes Ewing sarcoma is some sort of transitional or temporary cell that is not too easy to find in the wild. The sex ratio we've looked at by single year of age, and there is a male preponderance which gets greater during later adolescence. So that is some suggestion that there is a hormonal aspect to Ewing sarcoma risk, but not too many people have investigated that. And now we move on to what really is the biggest clue for Ewing sarcoma etiology, which is the very distinctive patterns by ancestry. In fact, Ewing sarcoma has some of the widest differences in cancer risk of any cancer. And so in the US and SEER, it's very clear that non-Hispanic whites have the highest rate of Ewing sarcoma, followed by Hispanic children who have roughly 2/3 the incidence. Asian kids who really, I think, is mostly driven by East Asian here, have about half the risk. And then there's a very low risk in non-Hispanic Black children, which has been noted for decades, but nobody really investigates it. A couple years ago, we decided to look at this question on a worldwide scale. using data from the Cancer Incidence on 5 Continents database. And you can see from our map that there is some countries in dark blue with high rates. They're not high overall. It's still an extremely rare disease, but comparatively high. And then in some places, most notably Central Asia, but also Sub-Saharan Africa, there's almost a complete absence of the disease. And so we took all the populations in the CI5 data set, and we just ranked the incidence. We also classified each registry or country by predominant ancestry, and then we had to come up with some statistical test, but this was wildly significant. But there's very big difference in incidence between the top ancestries and the bottom. At the top, to my surprise, was Polynesian. So it's too small for me to read, but Pacific Islander and Maori in New Zealand, New Caledonia, which I had to look up, but then also the Middle East and Europe. And then at the bottom is a number of countries in Africa which had no cases whatsoever reported in the CI5. And we actually had to go ask for help to see how to put a confidence interval around a 0 rate. You only do the upper confidence interval there. I'd also like to note down at the bottom here is China, which you can barely see the error bar because there were like 300 million kids in the data set, and yet next to no cases. So that's a remarkable finding, in fact. So another spot on the zebra is the widest difference in incidence by ancestry. Here's another question that nobody seems to be asking, which is, why do we have cognate cancers for almost every cancer in humans in wild animals? But I have not found, and I have searched, a single case report of Ewing sarcoma in a wild animal. Jaime? Nope. Yep. I've asked you over the years and still not a single one. So, that's just another spot on the zebra here. There's no animal cognate, and that's very unusual. So moving on to the analytic epidemiology. As is typical for rare diseases, people have done what I call kitchen sink studies. In other words, they just ask about everything under the sun to try and see what pops out. What else would you do when there's a rare disease that you know nothing about? At a high level, we looked in SEER data where they have a file that you can look at area level SES. And we just wanted to ask the question, if you look at SES versus ancestry, which one comes out of the wash? I can think of SES as sort of the compilation of all the exposures and behaviors that correlate with one's place in life. And what we found was that there was no association of SES, although maybe a hint of a increase in risk with higher SES, but not significant for Ewing sarcoma. But that controlling for SES, the ancestry differences in risk that we see in the crude data persist, suggesting that it is something about ancestry rather than SES or exposure. Also very oddly for a rare cancer, and when I say that there's only a few analytic epidemiologic studies, there may be fewer than 10, maybe even 5 or so. But unusually for such a small literature, there have been a couple of associations that have come out of the wash with some regularity. And so there's a hint that parental occupation is related to Ewing sarcoma, and specifically parental occupation in farm work or agriculture. And there's been nobody who's followed up on this since it was published many years ago. Speculation would be either that it's zoonoses from exposure to animals, or it could be pesticides, et cetera. Although without further work, which is next to impossible to pull off, we might never know. The other really oddball observation that came out of analytic epidemiology was an alleged association between hernias and Ewing sarcoma. Hernias are developmental defects, in which one organ is not held in place, and so it kind of invades another space, not in the cancer sense, but just in a physical sense, right? And surprisingly, there were 5 studies, and in the meta-analysis, it raised the risk. It's an odd scale here where the risk is going to the left. But here you see that there's roughly a 2 to 3-fold risk of Ewing sarcoma if the child had a history of hernia. And that one we have followed up on. So I'll tell you about that in just a little bit. There also is a virtual absence of family history for Ewing sarcoma, which is another aspect that is very bizarre. Most cancers have a fraction of cases that are due to high penetrance variation, which often appears to be familial, if it's inherited from the parents. This is data from the Swedish Family Cancer Database, which shows Ewing sarcoma is relatively unaffected with a family history, and similarly here. I kind of inadvertently demonstrated this several years ago when, with grad student Rianne Zimmerman, who's now faculty here with a new name, we did a catalog of genetic syndromes in childhood cancer. And here's what you get for ALL, just skimming the literature. There are dozens of reported associations between genetic syndromes and ALL. And here it's for hepatoblastoma, which is even more rare than Ewing sarcoma, but there's, again, over a dozen syndrome associations in the literature. And then here's what we got for Ewing sarcoma. 2 reports of coincidence in Down syndrome, 1 in XXX syndrome, 1 Leaf-Raumanni, 1 congenital optic atrophy, and 1 18q deletion. So, we didn't go through and calculate like number of syndromes by incidence, but clearly there seems to be a deficit of high penetrance syndromes that cause Ewing sarcoma. So another spot on the zebra is weak to no evidence for familial syndromes. Of course, we're now 10 or more years into the sequencing era. There is also sequencing error. And you'll notice that oddly, after what I just told you, it's kind of odd, but nearly 10% of Ewing sarcomas in the first big sequencing study in 2015 suggested that 10% of Ewing sarcoma had a high penetrance mutation in a cancer susceptibility gene. But once you broke down that information, it was 5 different ones, rather than here, adrenocortical carcinoma, 3/4 or more of them had a p53 mutation and at a specific spot in p53. Some further work from people looking at data in the Gabriella Miller Kids program suggested that FANC-C genes mutations were overrepresented in Ewing sarcoma. But honestly, if you look at the numbers here, it looked like maybe a 2-fold increased risk, which is not something that I consider high penetrance. In fact, that's much more along the lines of what we find with common SNPs in childhood cancers. Others have looked at whether there is variation in the EWSR1 gene, which is the fusion partner, whether there's some germline vulnerability to breakage and creating the translocation. And there was nothing that came out of this study, so that's been more or less put aside. On the other hand, people have successfully applied genome-wide association studies to Ewing sarcoma. When people started doing this, they were thought foolish because these are rare cancers. You don't get large sample sizes. And the word from on high from the adults is SNPs have very low association with cancer, and so you need a very large sample size to pull it off. Well, this paper came out in, I think, 2013, and you can see in this Manhattan plot that there's two very large hits in TARD BP and EGR2, which came out of a GWAS with maybe 500 people. So that was unusual, although not for childhood cancer as a whole. And some follow-up work looking at those loci have suggested that, in fact, they tag GGAA repeat regions, which are, again, sorry, I guess it's not again because I didn't mention it too much. Those are the germline kind of landing pads for EWS Fly1. And so the GWAS seems to have picked up the variation that determines the binding efficiency of EWS Fly1 and its ability to enact its transcriptional program. This is maybe shown quite a bit better in this paper, but it was this research that really turned me on to the fact that germline variation may be the reason that we see such different patterns of incidence in different populations for Ewing sarcoma. There was a subsequent follow-up GWAS, which also found more hits. And unusually, these are very strong hits. So just to give you an idea, these are 7 loci that describe over a 10-fold difference in risk according to the polygenic risk score. In prostate cancer, for instance, they have 150 SNPs, which together describe maybe a 5-fold difference in risk. And so this even made the abstract to this paper, Ewing sarcoma, for being a rare disease, seems driven mainly by polygenic risk with extremely large odds ratios per allele. By contrast, in prostate cancer, the top hit per allele odds ratio is maybe 1.2. So we just have a strange situation where these SNPs probably tagging GGAA microsatellite repeats are conferring a much larger degree of risk than anybody would have guessed. Here's how they stated it in the abstract. Additional research looked at low-frequency SNPs in the same loci and suggested that they too were associated with Ewing sarcoma. Again, probably they are tagging the variation we actually want to know about rather than being causative in and of themselves. So another spot on the zebra is the unusually strong association with common SNPs. And now I get to move more into research that we've done collaboratively. So I may be the only person who remembered that hernia was related to Ewing sarcoma. And I kept that in the back of my head for 20 years. And then a couple of years ago, noticed that there was a GWAS in the UK Biobank for hernia. And hernia itself is a heterogeneous set of diseases. You may have heard of diaphragmatic hernia, which means that a child's born without a diaphragm supporting their lungs. It's a very serious defect, right? Because if it's not corrected, the child can't breathe. And then there's much less serious ones like an umbilical hernia, which I think mainly means that you've got an extreme outie belly button that just needs to be corrected, right? And then there's inguinal and other types. Well, they produce these GWAS, and once you have SNPs related to a phenotype, you can do an integrative analysis to see whether there's any evidence that, Ewing sarcoma is related to hernia. So I'm inordinately proud of following this up 20 years later when nobody else knew or cared about it. But and this was work with Dr. Tianzong Yang in biostats. We took the UK Biobank data and correlated it with summary statistics from the existing GWAS for Ewing sarcoma. Here you just see and then we took it from a top level, like genetic correlation, genome-wide, and then to the individual loci. So I'm not going to bore you with all the minutia, but you can see that the hernias correlate pretty well with each other, probably because they are related defects in collagen or whatever parts of the body hold in other parts of the body. Although they don't on a global scale correlate very well with Ewing sarcoma. But once we looked at Mendelian randomization, which for those of you who aren't genetic epidemiologists is a fancy way of using SNPs to assess the causality of an association. All right, so if you think about each SNP as like a pull of a slot machine, some people will get 7 cherries, some people will get 7 lemons. Use this information to try and assess causality. For inguinal and diaphragmatic hernia, the kids who had those SNPs that raised the risk of those hernia phenotypes had 30 and 100% increased risk of Ewing sarcoma as assessed by genetics. And then digging into the specific loci, we found pretty strong evidence that the HMGA2 gene, which was one of the SNPs that came out of the GWAS for Ewing sarcoma, that it was not just causal in the statistical sense, but it was actually the same SNP that was causing both hernia and Ewing sarcoma. And so that's the co-localization test. All right, so moving back to the GWAS, I just put this figure together to explain that while we have GWAS SNPs, which by the way, I forgot to mention, were noticed only in, have been examined only in children of European descent so far. But we compared the SNPs to allele frequency in 1,000 genomes. And what we don't see is a clear pattern where these SNPs are more common in European genomes and less common in other genomes that have a lower risk. And so far the GWAS, while they've skimmed the cream off the top, they're not explaining these differences in incidence by ancestry. Another group has looked at mitochondrial DNA haplogroup, which obviously tells you about maternal lineage, but doesn't necessarily correlate that well with autosomal ancestry. And what they found was, not surprisingly, evidence that if you have a European-derived mitochondria haplotype, that's overrepresented in Ewing sarcoma cases. All right, well, here's some data that's still as yet unpublished, but still very interesting. I was part of Gabriella Miller Kidd's first study to sequence trios with Ewing sarcoma, and 49 of these kids out of about 400 were Latino, who are a admixed population. So we compared the risk SNPs that were found in Europeans to see whether they replicate in Latino children. We got controls, we matched them 4-to-1. Here's the PCA plot that just shows where people land. And what we found was that global European or African ancestry proportions were significantly different in Latinos. We had a little bit of question about matching because Latino as a umbrella term belies a lot of heterogeneity within the population. But what we found was that there was an inverse association with the percent African among Latinos, and then as you would probably predict from the pattern of incidence. And then interestingly, though, the SNPs that were associated with Ewing sarcoma in Europeans replicated and maybe even had a higher effect in Latinos. This is just depicting the GWAS loci. But the main thing to know here is that if we look at the polygenic risk score, where you add up the SNPs multiplied by the size of effect, we're actually getting a higher odds ratio among Latinos for the same SNPs that cause Ewing sarcoma or are correlated with Ewing sarcoma in Europeans. And so there's quite a bit of interesting follow-up work to be done there. Sorry, and for African ancestry, you can see that there was a significant lower risk with increasing percentages. And I'm just gonna skip that. So we do a lot of work in my division with birth registries and newborn dried blood spots. And so for the past 7 years, I have been approved to get the 30 years worth of Ewing sarcoma spots from California. For reasons well beyond my control, they've only sent me 1/5 of them. But once they send them to me, half of the cases are Latino, so we can expand this line of research, basically have a comparative genetic epidemiology with the European kids. And this is an analysis of the birth registry data, just showing that there is not a lot of differences that would, in the birth registry, that would explain the differences in risk. All right, so I already mentioned that the GWAS is really just probably tagging the underlying differences in sequence that we are really interested in. And the reason that we haven't gone any further is that looking at structural variation is not very easy to do with standard read sequencing. And so long read sequencing, which, so standard read is 100 base pairs. And if you have a repetitive region, it's very hard to resolve that, especially something like a GGA repeat, which the average number of repeats is 26, which means that the repeat length is over 100 base pairs. It just doesn't resolve very well. As opposed to using long read sequencing, which will give you a 10,000 base pair read. And what we see is that there are significant differences in the length of total genome due to structural variation in different populations. And so this is a global view. There are extremely large structural variations that is not really what we're talking about. In Ewing sarcoma, if we could have long read that just did, say, 500 base pairs, we could probably resolve this. But nobody came up with that technology. So we're talking about doing some massive overkill of long read sequencing to get at these kind of short microsatellite repeats. Earlier work from colleagues Steve Lesnick and Josh Schiffman, when they were both at Utah, showed that 2 of the EWS Fly1 response genes that were shown in some early ChIP-seq and RNA-seq experiments when they went and looked at the length of GGA repeats near these top hits, they found quite a bit of difference in length between African and European populations, which suggests that it may be just natural variation in these microsatellites that is driving the difference in risk by ancestry. And then there is a little bit more evidence here. So really, I set about to answer the question, why is it that we see such distinctive patterns of Ewing sarcoma by ancestry? And then that led to the question of, how do you study a disease that's virtually absent in some populations? And the answer is, you have to kind of create your own Ewing sarcoma in vitro. And so this is a collaborative project with Bo Weber. And honestly, one of the most gratifying projects that I've been involved with, because it is, I think, very different. And probably the proof of that is that Bo managed to pull off funding it on the first try at NIH. So I'm just going to tell you about the pilot work because the larger project is ongoing. So we had kind of two problems, right? One is that there are some ancestries where you just don't see a free-living, naturally occurring Ewing sarcoma. And so we decided to focus on lines with differing levels of admixture, focusing at first on African and European admixture, although we're also going to look at Latino lines. And then secondly, I already mentioned that the cell state is very important, and you need to have a permissive cell type. And so that led us to induced pluripotent stem cells, which we've differentiated into neural crest cells. And those are receptive enough to EWS-FLI1 that we can introduce it by viral vector, which has become the standard in Ewing sarcoma. Let that incubate for 96 hours, and then we get RNA-seq before and after, and then after we also do ChIP-seq or cut and run or cut and tag. I kind of didn't like it when they started naming these assays after Steve Miller songs. But in any case, we want to look at EWS-FLI1 binding. We want to look at the downstream transcriptional effects, and so on. And so in our pilot study, we managed to get lines with that were either 100% European or 100% African, and then some levels in between. And the first thing you see is that the neural crest cells tolerate EWS-FLI1 expression for EWS-FLI1 introduction long enough to get a readout, although they tend to die after a while, even in the NCCs. We didn't bother trying to introduce it. I don't think we did try to introduce it to other lineages to prove to ourselves that this was necessary. But in any case, one question that study section had was whether these cells would survive long enough to give us a readout. So we had to prove that to them. We got similar expression profiles at 48 and 96 hours, and then we ended up going with 96 hours for reasons I no longer remember. But here's the really interesting thing. So we do a PCA plot of RNA principal components of RNA-seq, and you see very clear separation between wild type and the same cells after introduction of EWS-FLI1. So that's good. This mutant transcription factor, which has a pernicious program, has transformed expression in these cells. Of course, what we were looking for was a kind of a clean difference in the other PC, which would have been ancestry, and we found some hint of that, but not nearly as clean as you might think, albeit it was enough to justify the grant. Higher European ancestry in the pilot work resulted in a prolonged maintenance of EWS-FLI1 expression by this GFP reporter. And so that was consistent with our hypothesis. And then you can see it here, highly significant. And basically, the more European ancestry, the longer these cells survive, the more African ancestry, the less they did. And then when we looked at the transcriptome compared to using GSEA, gene set enrichment, we were very gratified to see that Ewing sarcoma transcriptomes were, it was most similar to Ewing sarcoma transcriptomes, which is exactly what you'd expect, so, or what you'd hope for. So we think that we have kind of validated this model as the early, to tell us about the early molecular events when the EWS-FLI1 is present in a cell. The next step was to try and see whether there were loci that were responsive to EWS-FLI1 differently depending on ancestry, and we found quite a few of those. And when we looked at transcriptome, and again, this is in a small number of samples, they did seem to cluster by ancestry with higher African ancestry on the left and higher European ancestry on the right. If we look at occupancy, meaning binding of EWS-FLI1, it was a similar story. And so our goal then was to expand this, and we're doing it now, to find, and this was my major contribution to the research, an acronym, ancestry-linked differential EWS-FLI1 response, or ALDR loci. And so we are doing RNA-seq before and after introduction of EWS-FLI1, cut and tag, attack-seq, and then long-read sequencing, because ultimately we're just using ancestry as a proxy for the length of these microsatellite repeats. If there were collections of iPSC with long-read data already available, we might just go straight to the GGA repeats, but that hasn't happened yet. And so we're looking for ancestry-linked differential expression, differential binding, changes to open chromatin within 1,000 base pairs of the repeats, differences in mean repeat length by ancestry, and I think we also threw an ETS sequence variance associated with ancestry. And we take those, and I say we, I mean Lauren and Rachel, and turn those into our candidate loci. We had preliminary ALDR loci showing both differential expression and EWS-FLI1 occupancy. And now those loci, because they seem to drive the very large difference in risk between ancestries, those are now candidates for developing an intervention or therapy. So Bo, and this is now well beyond my capabilities, you know, is knocking them out, left, right, down, to see if messing with those genes improves the ability to kill Ewing sarcoma. Of course, as Hillary Clinton said, it takes a village. I just want to specifically thank several people, and including Sampi Mishra, postdoc in Bo's lab, who's been taken this project and run with it. Dr. Lauren Mills in my group, who's our lead bioinformatician, as well as almost Dr. Rachel Moss, who's soon to graduate, who did a lot of the bioinformatics for the pilot study. And then James Dutton, if you are out there online watching this, I apologize, I should have put your picture up too, because James is the iPSC whisperer who made this project possible. So on to the future. I continue to be near obsessed with this tumor. I'm going to, I may actually send somebody out to California to grab the spots that I've been owed for 7 years in person. And so this will be a study, focusing on admixed genomes and then integrating our molecular data from iPSC. We will be expanding the iPSC work to include more African-American lines, but also Latinos. And going back to that first graph I showed you, if anyone knows where I can find iPSC from Polynesian donors, that would be incredibly interesting. And then lastly, like I said, we've been using Ancestry as a proxy for microsatellite variation, but we could just go straight to the microsatellite variation. Oxford Nanopore is now about $1,000 for a whole genome. And so it's moving into the realm where we might think about a population study. So, I will someday propose to the Children's Oncology Group that we simply do long-read sequencing in case and control sets. So I have a lot of people to thank at the U, especially Bo, for being interested in this question and, doing an amazing job at, bringing this to fruition. Did I mention that he got the NIH grant on the first try? Lauren and Andy in my lab for the excellent informatic and population genetics help. Tianzong, who's also taken a strong interest in this very rare cancer. Rachel Moss, Kelsey Becklin, who started the project, the hands-on part in Bo's lab, and Sampi, who's taken it and expanding it to the much larger. At the National Cancer Institute, Mitch McKeela, this is his baby. He was handed it by Stephen Chanik. I was also incredibly happy that our former doctoral student, Aubrey Hubbard, is now his postdoc and working on the next phase GWAS for Ewing sarcoma. So we continue to collaborate with her. At Nationwide Children's, Stephen Lesnick, who is kind of the American king of Ewing sarcoma biology, about to retire. And then his successor, I think, Emily Thiessen, who's been collaborating with us. And then also Josh Schiffman at the University of Utah. And with that, I left a few minutes for questions. Thank you.
I have microphones for anyone with questions.
What do you think explains the really low incidence?
Hang on, Bo.
I'm sorry. I was just interested the low incidence in China. I guess we hadn't talked about that, so I'm kind of curious what your thoughts are there.
I mean, once people started looking at structural variation by long read, it actually blew my mind that humans' populations can vary in the length of their genome by up to 10 or 15%. So structural variation is the dark matter of the genome that we really don't know about right now because we haven't been able to measure it very well. That's ending, and soon it will be revealed. But we know specifically that GGA repeats are the landing pad for EWS Fly1. So, I mean, my strong supposition is that for whatever reason, Chinese population has a different structure of GGA repeats that just happen to be less amenable or less permissive to EWS Fly1. We didn't actually talk about the other hypothesis that we probably can never test, which is that EWS Fly1, the translocation, appear at different frequencies in different populations. I don't know how you would ever look at that because, if it just appears in a rare cell type that's transitional and that you could only find in if somebody dies, then it's just never going to be found. Yeah.
Hang on, I've got a microphone. Thank you.
Logan, thanks for the for the nice class for you in sarcoma. So about the, I probably missed that part. How did you select for neurocrest cells to express CWS-FLY1?
Oh, save me. He wants to know how did we didn't select for them, but took them from pluripotency, and James has a cocktail that walks them forward. And that's literally as much as I could tell you.
Okay, but that population probably is not homogeneous 100%. And then when you do transcriptional analysis and all of the other things, probably you get some miss signal, or you get a signal from different non-permissive cell populations or cell types that are.
Can you hand the mic to Lauren and she'll answer?
So the differentiation actually works pretty well. 80+% of the cell population is really strong for the markers that SAMP tests for. So it's a pretty homogeneous starting population. We don't get expression data from cells that die because they're dead. So at least I am pretty happy with the level of homogeneity of the population, but it is something that we looked at starting for.
Our typical transduction rates are 80+% with the virus, and so weighing all of sort of the logistical requirements of doing this in so many lines, we just basically said we're going to basically generate a very high titer, high quality virus and set a threshold cutoff. For gut and tag, it doesn't matter because it's an antibody that detects the protein. So if it doesn't express the protein, it's not a question. We need to be very high level.
I was more concerned about.
Hang on, guys. So we have a virtual audience, so it's really hard to just have an open discussion. So because we've got no mics in the ceiling. And so I'm just going to move to this question with Doug, and then if you'd like me to come back with the microphone, I will.
Yeah, we'd love to have a microphone. So in some diseases, like breast cancer, where there are a lot of them, the polygenic risk score, odds ratio, hazard ratio changes by age. I know there's not a huge age distribution in Ewing, but does it make a difference? Age of
The GWAS so far, the larger one that I presented was 700 people. And I don't think that, they've divided it yet. But like I said, our former pre-doc is now postdoc at NCI, working on the next phase GWAS, which is like 1,500 people. I could ask her that question. Yeah.
Great. Any other questions today? Let's say thank you to Dr. Spector one more time.
Thank you.